So instead of testing on animal trials and testing on humans, what we have is we have several wells with cells in them with cardiac tissue. We call it cardiac tissue, and we call the whole thing a heart on a chip. Okay, so what we do is, for example, I want to see if this drug is gonna help slow down the rate of beating, or is it gonna pump it up. So I apply the drug in every one of these wells, and then I can connect these little gold electrodes to the computer, and I can see if it's going faster or slower. Take a look at this around, and then I'm going to show you the cells, how they look under the microscope. So in those things, we will have like a layer of cells beating. So this is going to be the layer beating. This is cardiac cells from stem cells acquired. You see how they beat?
These are real samples from...
This is real samples under the microscope. I wish I had some. I have some growing right now in there, but they're not old enough to beat right now. It takes another 7 days for them to beat. Otherwise, I would have showed them under the microscope. So this is what we got. So let's say this is beating... How much is a human heartbeat per minute?
What is it
Average. 60. 60 is really good for someone who's like an athlete. Technically, like along all people, it's about like 70, 75. So let's say that this is 75 beats per minute. Now I put a drug in those wells, and then I monitor the beating, and if it goes much faster, then that's gonna be a good drug for someone who has like a very slow heart rate to increase it up and reverse. Do you see the reason why we're doing this? Yeah? Any questions, guys? Is it clear? Yeah? So this all happens with biomedical engineering. If you go into biomedical engineering, because it's an interdisciplinary major, right now the sciences are so much and so big that we need to condense everything into one thing and then try to add that into people. So biomedical engineering takes all the information from material engineering, from let's say electrical engineering in our case, and then it combines all, and biology, and it combines all into one concept of biomedical engineering that we can apply into these things like that.
Oh, yes.
All right?
What happened to you?
Any questions? No? Do you have any questions with the setups over here? You can go around. This is like other stuff that we have over here. We use also microelectrodes.
Can you show the guys where you grow the cells? Where you have the cells that in 7 days might be
yes.
beating?
You want to see the cells, you mean?
Whatever
Okay.
you can show.
All right, sure. Come over here. You guys can stand on that side. All right? Now I have one recording right now. You saw, excellent, that's an incubator. What is an incubator for? Why do we need an incubator for cells? Excellent, because your body is 37 degrees. We can't just leave them outside in this weather because they're gonna be frozen and die, okay? So this is 37 degrees and 5% CO2, which is the best environment for them to grow. Let me grab this. So this is a fully sterile environment. So this is a fully sterile environment. So every time that we want to go in and out, we have to spray our hands with ethanol to make sure that everything is super sterile. We make the cells under that, and then we put them inside here for growing. So what I wanted to show you was a recording that we're doing right now. So you see that device in there, the chip that I showed you gets connected to that thing, all right? And then we close it. Every one of that, you guys can move here more to see it. Every one of those little gold ones goes inside of that. They have 8 pins that reads from, 8 channels, and each of the channels have cells inside them beating right now. So, not beating right now, probably they're growing. So I did them yesterday, I cultured them yesterday. This is results from 8 channels. It's saying that if it keeps going up, this is resistance. So it's like some sort of an electrical readout. It says that the more cells that gets attached, the more this line goes up. So I can see how well this is. So I can say like these wells, they don't have that much of a good cell attachment. This one is doing pretty well. Okay, and then after 7 days, it's gonna start beating, so it's gonna be like a rhythmic signal, like the EKGs that you guys see outside. All right. So by the way, what to expect from cells is just a solution. So what you're gonna see without microscopy is just like a red solution in there. If you can see the device, I really can't take that out. But do you see that thing in there? So that technically is all the cells are attached on the bottom, and then that's the food technically. We call it media. So it has glucose, glutamate, everything that they need to grow. All right. Any questions? No?
Can you explain a little bit how's the extraction process? How you obtain the cells?
Sure. So there's two methods. We either get it from rat hearts, but because we want to go towards the fact that we don't harm animals anymore, we take them from patient's own blood. So we get blood samples, human blood samples, and there's like a process to extract the cells from there into like stem cells. We don't do it ourselves, we give it actually outside to give us the stem cells. Once we got the stem cells, then in a process of 11 days, we put chemicals in to get them attached. We put chemicals in to take them transformed engineering into cardiac cells specifically. And then finally, after 11 days, we have them beating into cardiomyocytes, which are heart cells. All right?
Can I ask you another question?
Of course.
After you have beating cells, what do you do with them? What are you guys taking this experiment to?
Okay, that's a really good question. So we, right now in my project, we monitor how fast do they beat without anything invasive. So optically using a microscope and using different techniques like SPR. We monitor how they beat, how fast, what is the frequency. We monitor how the effect of drugs are on them. For example, we put blebistatin. Blebistatin is a drug that stops the heart rate. So in cases in like fibrillation, you add blebistatin to stop the heart rate and then restart it again. And then see ATP effects. ATP increases heart rate, so we do that. Then we monitor how much are they using oxygen, so respiration, because it's an important part of cell growth. So we monitor oxygen. And also we monitor how do they eat in terms of glucose, lactate, and fatty acids. Those are important things for cell to grow. So we monitor if it drops, we give them more. If it goes up, we decrease it. So we have everything of that under control in that little chip. Cheap, fast, ethical. We don't need to kill 1 million animals anymore. We don't have to go through devastating human trials.
And also, is this for medication, create medication?
And it's exactly, the target is create medication. Faster medication that takes to market. Every drug right now takes 20 years to go to the market, if they ever do. 20 years. With this platform, that will decrease it at least by 50%.
So a little less so.
So this basically to cure heart diseases.
It is to cure heart disease, yes. So we are going towards, it's not impossible, it's like very possible in the very near future actually. We would assume 20,
More large scale.
2050 to be safe. That there's going to be many drugs left and right coming out that you don't need to do surgery. There is nano designed drugs that can just go and electronically control, that can start regeneration and treatment and everything.
Cool. Heart. So we don't need a heart transplant.
We don't need a heart transplant. We can start. That's actually another thing that the tissue engineering part are doing. We're not focusing on that, but that's another huge area of biomedical engineering, that they start making stem cells and then they take it into hearts.
Only hearts or regular?
No, I wanted to actually say that. No, the same thing applies, because concept number 1 was nanotechnology. Concept number 2 is tissue engineering. So it can be kidney, brain, lungs, and the kidney, brain, lung samples are actually available right now at Wyss Institute at Harvard. They're making all of that chips, heart on a chip, brain on a chip, bone on a chip, lung on a chip, and then they put the whole thing in a little printer called human on a chip. So when you apply drugs, it goes through all of the systems, and in the computer you can say, oh, okay, this is toxic for lungs, so we don't do that. Oh, this is toxic, it doesn't help going through the renal system, the kidneys, so we don't do that. Oh, this is better for the heart, and it doesn't affect the brain or the kidney, so let's go with this drug. So this is hardware necessity. So there's so many cool stuff happening in biomedical engineering, and it's just keep going up. There is no degradation for at least another 100 years because we just starting at this stage. All right?
One more question.
Yeah, please.
Yes, go ahead.
Let me ask you, what software do you use to analyze this data?
Many softwares, many things. So LabVIEW is a thing that if we want to develop our own software, we use it. MATLAB is another one. ANSYS is another one. Python is another one. So it depends on if we want to write the code ourselves or if the software comes with the devices that we buy. If the software comes, it's just there. But if we're writing it, we either use Python or we use LabVIEW or R. It really depends on what is the application. All right? Questions, guys?
It's an important thing I read about. They already invented something to grow the tooth with
Oh,
stem
really?
cells.
Really?
Yes,
I didn't know that.
it's on the market. I mean, they're testing it, so you don't have to have an implant. They're going to inject something with stem cells to make your tooth grow.
That's very nice. That's very nice. The point is, it's just not harming anything. They're extracting it from the very basic. Bye. Thanks for coming. Yeah, you're just extracting it from patients' own bloods, bone marrow, anything, non-invasively.
Yeah, I hope they do it soon, so we don't.
I really hope
we don't
so,
get
yeah.
implants.
It's exactly something that wakes me up every morning. Honestly, this is very promising, and I do believe in it 100%. All
So
right.
that's why we need our future generations to get involved.
We do need you guys, yeah. We need you
To
to
get
pass
involved
along
in-
this. You guys have, the point about you guys is that you don't have the restricted mind that people get when we go so much into academia after a while. You can think outside of the box. So when you know these stuff up in the front, you can daydream about it. You can like imagine, oh, what would I do if I was there? Oh, what could have idea can come to your mind to apply in this that cannot come to my mind? You see, it doesn't matter if it's feasible or not feasible. It's gonna be feasible one day. All right. The fact that it passes your mind, it's really worth it.